Hello, my name is Chimi. I'm currently taking the BIOL110 class at DKU. So I will be presenting on the topic of bacterial transformation. This is a fundamental technique in molecular biology used to introduce foreign DNA into bacteria. And this is actually the context of the fifth lab in my course. This procedure allows us to study gene expression, protein production, and explore how bacteria adapt to new genetic materials. For learning objectives, the primary goals of this lab were: the first is to understand the key concepts such as plasmid, competent cells, and cells transformation. The second is to perform a bacterial transformation using DH5 alpha competent cells and PUC19 plasmid. The third is to plate and incubate bacterial cultures and observe the resulting colonies to analyze whether the transformation is a success or not. Before starting the experiment, you need to understand some background information. In this experiment, we are going to use the PUC19 plasmid, which contains the AMPR gene. This is a gene for ampicillin resistance, and also the LAGZ gene, which encodes the beta-galactosidase. These two components enable us to identify transformed bacteria by the expression of beta-galactosidase, which will produce a blue color in the presence of X-gal. And the things below are the materials we need to use during the experiment. Next, I'm going to introduce the procedure of my experiment. So in the preparation phase, DH5 alpha competent E. coli cells were thawed on ice, and two 1.5 millimeter microcentrifuge tubes were labeled, and one as plus for cells with plasmid, and the other as minus for cells without plasmid. The corresponding LB, AMP, IPTG X-gal and LB, IBTG X-gal agar plates were also labeled. And next, 50 microliter of competent cells were transferred into each labeled tube. 10 microliter of PUC19 plasmid was added to the plus tube, while the minus tube remained without plasmid. The tubes were then incubated on ice for 20 minutes, followed by a heat shock at 42 degrees for 45 seconds, with immediate cooling on ice for 2 minutes. And finally, 500 microliter of LB medium was added to each tube, and the tubes were incubated at 37 degrees for 20 minutes to allow the bacteria to recover. And this is a graph about how the transformation processes. For plating and incubation, 100 microliter of each sample was plated onto the corresponding agar plates, and glass beads were used to evenly spread the bacterial cultures across the plate. The plates were then incubated at 37 degrees overnight. And for observation and analysis, we will do this on the next day. We will examine the bacterial growth, and we will analyze the growth patterns as well as colony color to determine whether the transformation is success or not. This is the expected results. We should see the blue colonies for the plus LB-AMP group, lone for both LB groups, and while there should be no colonies for the minus LB-AMP group. This is my result for my own experiment. The experiment, as you can see, the experiment is quite successful because my results align with the expected results. For the LB-AMP plus PUC19 group, I can clearly see the blue colonies on the agar plates. For the LB plus PUC19 group, I can see bacteria on the agar plates. I was expected to see some blue colonies, however, it's not obvious. Maybe it is because the number of the white bacteria is much bigger than the blue bacteria. For the LB-AMP minus PUC19 group, the surface of the agar plates is still clean. No bacteria can be observed on the plate. And for the LB minus PUC19 group, it is quite similar to the LB plus PUC19 group from the outer look. The presence of white spot bacteria was also clearly observed. This experiment successfully demonstrated a bacterial transformation using the PUC19 plasmid, and the transformation was confirmed by the presence of blue colonies on selective media, allowing us to identify bacteria that took up the plasmid. For the real-life application of the experiment, uh, we can use this technique to produce vast quantities of one protein. So how can scientists use transformation to do this? First, scientists should insert the gene encoding the protein of interest into a plasmid, which is then introduced into bacterial cells. Once the bacteria take up the plasmid, they use their cellular machinery to express the gene and produce the protein. Also, as the bacteria grow and divide, they replicate the plasmid, resulting in many copies of the gene and vast amounts of protein. Actually, this method is already widely used in biotechnology, produce proteins such as insulin and enzymes. Last, I want to thank Professor Lee for his excellent teaching. I also want to thank Su Hua and Mian Mian for their patient guidance in the laboratory. Thank you for listening.